Under mutation and selection technique or modifications, we have discussed cellular permeability, modification of cellular permeability. Second, isolation of auxotropic mutants as a method for strain improvement for the overproduction of microbial metabolites. Then the third type of modification is isolation of mutants that do not recognize the presence of inhibitors or repressors. The difference between second modification and third modification is that in second modification, the inhibitors and repressors are not produced. But in third type of modification, inhibitors and repressors are produced, but their excess level is not recognized by the control system. That means the enzymes, key enzymes which are sensitive to inhibiting or repressing levels of metabolites, it is vanished or it will be lifted. The control will be lifted. The auxotropic mutants are basically useful for the production of intermediates of unbranched pathway, but obviously such mutants are not suitable for the synthesis of products which control their own synthesis independently. Say for example, this is the biochemical pathway, that is unbranched biochemical pathway. So in this case, auxotropy is the possible solution for the overproduction of intermediates like C and D. Whereas if you think about overproduction of E by auxotropic modification, so it is there is no possibility of auxotropic modification for the overproduction of E. Then what is the solution? The answer to this problem is to modify the organism such that the first enzyme, that is key enzyme in the pathway, no longer recognizes the presence of inhibiting levels of the end product E. That means the organism is modified, the organism is mutated such that now it will start production of enzyme, that is the first key enzyme, and that enzyme is a modified enzyme, and that enzyme is resistant to the levels of E. That means that enzyme is now not recognizing E as a repressor or inhibitor. So the site of modification will be related to the synthesis of this key enzyme. This is how this control will be lifted, and there is a possibility of overproduction of end product of unbranched pathway. So particularly this type of modification we can plan in relation to the microbial metabolites of this kind of nature, that is end products of unbranched pathways. So the isolation of mutants with changes in the recognition of control factors can be achieved by two types of techniques. First one is the isolation of analog resistant mutants, and second one is isolation of revertant mutants. Isolation of analog resistant mutants. Let us understand what is this analog. Analog means it is a compound which is very similar in structure to the another compound. Another compound means microbial metabolite. Say for example, microorganisms are producing lysine. Then there is a lysine analog called as AEC. So this lysine analog is structurally similar to lysine, but the only thing is that these analogs are toxic for the cell. That means the organisms will die in presence of these analogs. So that is the difference. So analogs are the similar, structurally similar compounds with that of the naturally containing microbial metabolites, but at the same time you need to remember that analogs are analogs are toxic for the microorganisms. Analogs of amino acids and nucleotides are frequently growth inhibitory, and their inhibitory properties may be due to a number of possible mechanisms. Now, here we are talking about site of toxicity or the mechanism of toxicity that leads to the death of cells. Toxicity of analog, it may be due to several mechanisms. One mechanism is that analog may be used in the biosynthesis of macromolecules, resulting in the production of defective cellular products. Say for example, amino acids. Amino acids are used in the making of proteins. So if instead of that amino acid, analog of amino acid is added to the medium, then that analog of amino acid will take the place of that amino acid, and it will get incorporated into the protein, and that protein will be a defective protein, and due to production of defective protein the organism will die. So that is the one mechanism. Say for example, in this diagram you can see the unbranched pathway that leads to the production of a growth factor called as E. For example, E is a primary metabolite, that is amino acid. So what is the regular function of E? The regular function of this amino acid is that after the synthesis it will go and it will build up a complete protein which is functional. This is what happens in normal case. Sometimes if there is any analog of this amino acid which is toxic for the cell, so what will happen? So wherever this E goes, for example, E star is the analog of this E. E is natural control metabolite and E star is its analog. So wherever this E goes, E star will go. For example, it will also get accumulated into the formation of protein. However, this protein will be a defective protein, and defective protein will lead to, so we can call it as it is a site of toxicity of analog. What is the site of toxicity? The site of toxicity is it gets incorporated into the cellular components for the formation of defective products, and ultimately it leads to the death of cell. This is the one mechanism of toxicity of analog. Another mechanism of toxicity of analog that may be responsible for overproduction. Why we are studying the toxicity of analog? Because this is something related to the overproduction. Another possibility is that the analog is not incorporated in place of natural product, but it may interfere with its biosynthesis by imitating its control properties. So, in the previous diagram, we have seen that this E is an amino acid and which is responsible for the synthesis of protein. So, that is the one target where that E goes naturally. Another target of that E is that it goes to the first enzyme. It controls the first enzyme by feedback inhibition or feedback repression. That is the second target of that natural control metabolite. So there are two things that this amino acid is doing, that it is getting incorporated into protein and it is responsible for controlling its own biosynthesis by feedback control system. Returning back to the same diagram, this E is responsible for the controlling the first key enzyme, and if this E star is the analog of this amino acid, then as we have seen earlier that wherever this E goes, the E star will go. But in this case, this E star is targeting the key enzyme. Okay, the E star analog of E is targeting the key enzyme. And this, what is the difference between inhibition by E and inhibition by E star? The difference is that inhibition by E, which is the natural amino acid or natural control metabolite, so it will go and it will inhibit or repress the first enzyme, but this inhibition or repression is reversible. Whereas if E star goes in place of E, that means if analog goes in place of normal control metabolite, it will go and bind to that first key enzyme and it will permanently inactivate that particular enzyme. That is the difference between the control by E and toxicity by E star. So ultimately, due to permanent damage to the enzyme, the cell is going to die. This kind of mechanism is very important if we are looking for the overproducers of E. How? So in earlier case, the site of toxicity was different, and in this case, the site of toxicity of analog is the site of control by normal control metabolite. So by mistake I haven't, the second box has to be, so the first box is site of toxicity of analog is, the second box is site of control by normal control metabolite. So that is very important, and if these two sites are same, then and then only there is a possibility of overproduction of that end product of unbranched pathway. Say for example, this pathway, so this organism is exposed to mutagenic agent. So this shows the biochemical pathway which is existing in the microorganism. A is converted to E through several steps. The enzyme responsible for conversion of A to B is the key enzyme, which is controlled by normal control metabolite, that is E. So when this organism is exposed to mutagenic agent, what we propose, what we plan, what we think, what we predict is that the possible site of mutation is this, which is indicated by the arrow. That means the possible site of mutation should be related to the synthesis of this first key enzyme. That means mutation has to occur in the genes which are responsible for the synthesis of this first enzyme. So why this will happen? Because we have seen that the site of toxicity of this analog is this enzyme. Therefore, the organism will try to modify its system. So what modification the organism will do? It will modify its enzyme so that it will get converted into a resistant variety and it can grow in presence of that analog. That is the strategy. So the possible site of mutation should be related to the synthesis of this key enzyme. So what will happen then if the mutation occurs at correct site, means the genes responsible for the synthesis of this enzyme, then what will happen that this mutant will start production of or it will modify its genetic machinery. That means it will modify the genes which are related to synthesis of this first enzyme. And now you can see the enzyme is modified. The mutant is shown at the bottom, and this mutant shows this biochemical pathway in which there is a red color star. You can see that is enzyme. Now earlier it was black color star, which was the natural enzyme, but after mutation now the enzyme is modified. Now what is that modification? The modification is that this enzyme is now no more sensitive to the levels of E. What is this E? E is normal control metabolite. You see, this is the desirable modification. Now this enzyme is resistant to E, and therefore if it is not recognizing E, it will also not recognize E star. What is E star? E star is analog. So this enzyme is resistant to the levels of E. This enzyme is resistant to E star also. This organism will be called as analog resistant mutant, which will be a overproducer of E, that is the end product. That is the strategy which is, so in this case we can see that the mutant is synthesizing an enzyme which is resistant to analog, as well as if it is resistant to analog it will show resistance to E. This is how this control will be lifted, and there will be overproduction of this end product of unbranched pathway. Then, methods of isolation of analog resistant mutant. What will be the medium used for isolation of analog resistant mutant? Of course, the medium containing analog. So, the mutation treatment is given to the microorganisms, and then the survivors of mutation treatment are transferred onto medium containing analog, different concentrations of analog. So whatever the microorganisms that will grow on this medium containing analog, it will be called as analog resistant mutants. So there is a method for isolation. This method is called as gradient plate technique, because we don't know exactly what should be the concentration of analog to be added in the medium. So the solution for that is gradient plate technique. So this is the plate in which the solidified medium, it is poured into plate in slanting position, and that medium contains analog. So analog containing medium is poured in the plate in slanting position, and then after solidification this plate is then further overlaid with medium without analog. So this will lead to formation of a gradient. Gradient means varying concentrations from left to right. If we see from left to right, what will happen? The analog which is present in the medium at the bottom, it will diffuse at the top. It will diffuse at the top. So if we look at the distance, that distance of required for the diffusion of this analog from bottom to top, it increases when you go from left to right. Therefore in the left zone there will be a high concentration zone, because analog can diffuse faster at the surface. And right side zone, right side plate, this is a side view. Right side plate will have a zone, a low concentration zone of analog. This is the front view that you can see. So after incubation this may be the picture. So left side is the high zone, which is high concentration zone, which is having high level concentration of analog, and right side is having low concentration of analog. So obviously on the right side there will be heavy growth due to low concentration of analog, and on the left side there will be a limited number of colonies formed due to high concentration of analog. So these are the possible analog resistant mutants, but mind well that all analog resistant mutants may not be overproducers. Therefore, the next step should be the resistant isolates should then be screened for the production of desired compound. This is very important. Now let us see an example of the use of analog resistant mutants for the production of primary metabolite. Sano and Shu, the Japanese scientists, they isolated a lysine analog resistant mutant of Brevibacterium flavum for the overproduction of lysine. In order to understand the mechanism that how lysine analog resistant mutant could be the overproducer of lysine, we need to go back to the biochemical pathway that we have already studied. Lysine belongs to aspartate family, and the biochemical pathway that we have studied in case of Corynebacterium glutamicum in the first type of modification. So same type of biochemical pathway it exists in Brevibacterium flavum. So the control of biosynthesis is like this, that which is similar in Brevibacterium flavum, although the title is different. You can see that aspartate is getting converted to aspartyl phosphate with this enzyme, that is aspartokinase. Aspartokinase, it is a key enzyme. We can see that aspartokinase is sensitive, or it is inhibited by concerted feedback control with lysine and threonine. So lysine and threonine are feedback inhibitors or feedback repressors for this aspartokinase enzyme. So what is this mutant which was developed by Shu and Sano? Lysine analog resistant mutant. So the lysine analog resistant mutant, this is the analog for lysine, that is amino ethyl cysteine, AEC. So you see that lysine and threonine, they are responsible for concerted or combined inhibition of the first key enzyme. So, and this analog, that is AEC, this can inhibit this aspartokinase in presence of threonine. That means this AEC, it will take the place of lysine if added in the medium, and this AEC plus threonine, it will target the aspartokinase. But what will be the difference between inhibition of aspartokinase by lysine plus threonine and inhibition of aspartokinase by AEC plus threonine? The difference will be that inhibition of aspartokinase by lysine plus threonine is a reversible process, whereas inhibition of aspartokinase by AEC plus threonine, it will be a permanent damage to aspartokinase, and this permanent damage to aspartokinase will leads to the death of cells, because now the cell is not able to synthesize any of these amino acids. So ultimately it will lead to death. This is how the toxicity of AEC is due to the inhibition of permanent inhibition of aspartokinase. That means the site of toxicity of AEC is the site of control by lysine. This is very important. So they demonstrated that AEC completely inhibited the growth of Brevibacterium flavum in presence of threonine, as I mentioned, but only partially in its absence. Why? Because aspartokinase is inhibited only in presence of two metabolites, lysine plus threonine, AEC plus threonine. So therefore, presence of threonine is essential. Also, this inhibition, it can be inhibition by AEC and threonine. It could be reversed by addition of lysine. So if lysine is added in the medium, this inhibition is reversed. What this suggests? This suggests that the target of AEC is aspartokinase. This is how it can be concluded that the site of toxicity of AEC is the site of control by lysine, that is aspartokinase. So this evidence suggested that inhibitory effect of AEC was due to its lysine-like effect. Lysine-like effect means its control mechanism of concerted inhibition of aspartokinase. So this AEC, what will be the method for isolation of AEC resistant mutant? Then you will say that the medium containing AEC. Wrong. The medium should contain AEC plus threonine, because the inhibition occurs in presence of, not only in presence of AEC but in presence of AEC plus threonine. So therefore AEC or analog resistant mutants were isolated by plating the survivors of mutation treatment on minimal agar medium containing AEC and threonine. So whatever the organisms that grows on this medium containing AEC and threonine will be the possible AEC resistant mutant and possible overproducers of lysine. So investigations of lysine producers indicated that their aspartokinase is not sensitive to the concerted inhibition by lysine and threonine. That means the organism is modified and its aspartokinase is modified so that this aspartokinase is now no more resistant to AEC, as well as it is no more resistant to lysine. If it is it is no more sensitive, this aspartokinase is no more sensitive to AAC, or it is no more sensitive to lysine. If there is no sensitivity, there will be overproduction of lysine. The question arises, why there will be overproduction of lysine only? What about threonine and other amino acids? So you can see in the picture, earlier picture, biosynthetic pathway, that there will be a control which is still existing over synthesis of threonine. Independent control is there, synthesis of methionine, independent controls are there. So there will be no excess synthesis of other amino acids. There will be only synthesis of lysine in excess.